Hello, this video is produced by the Video Records Project of the UC Davis Emeriti Association. I am Joan Borkman. The Video Records Project was begun in 1992 and since then has recorded over 400 interviews with emeritus and emerita faculty, as well as leaders from the campus administration and staff. These are the people who created the history of the campus. The interview you will see here is part of an archive of oral histories, first-hand accounts of the individuals and groups who worked to make UC Davis one of the nation's, even the world's, leading universities. Professor Emeritus Raymond C. Valentine was appointed in the Plant Growth Laboratory, where he had a distinguished career. He was also the scientific founder of Calgene Incorporated, a research and development company at the leading edge of biotechnology. His scientific interests involved the use of reductionism to address problems of fundamental scientific and societal importance, especially agricultural productivity and aging. Some of his scientific accomplishments include the discovery of ferredoxin, the identification and naming of the nitrogen fixation genes, and the development of Roundup resistance in crops. He is interviewed by his son and collaborator, David Valentine, professor of earth science and biology at the University of California, Santa Barbara.
So this is really exciting for me. My entire life I've been hearing stories about your childhood and your science and your education, and it's really great for me to be able to sit down with you and talk about this on camera, because I think the first time we've ever formally recorded anything, any of your stories, which you have many of. So I want to start today talking a little bit, going through first your childhood before we get onto your career and your science. So maybe you could start telling us a little bit about your childhood, where you grew up, and your family background.
Yes. Well, it's unusual. There was another famous Illinois person who grew up in a log cabin, but we had a very modern log cabin, except it didn't have outdoor plumbing, and in the wintertime, the chill was 33 degrees below zero, so it was a little different, but we got along fine. I had 6 brothers and sisters, and we all worked together, but no light, no electricity. We didn't have electricity until 1942.
And you were born in 1936,
so
until you were 8 years
so
old.
We didn't have electricity either. So the dark came at about 4:30 in the wintertime. So we had no TV, obviously, or no radios or anything. So my dad was a storyteller, and in those days it was all storytellers were the people who recorded and told us that we'd beg him every night that he's usually too tired, but we'd beg him to tell us a story. I won't tell you some of my favorites.
Yeah. And he was born in the 1850s, so he must have had a lot of stories.
He was born in 1881.
1881.
And so sometimes he would tell us stories. So I really enjoyed these stories. But at 2, I was my mother had my next sister, Barbara, and she was very hard to take care of all these kids, and so she had a person come out to help her a little bit, and I was called in for supper and didn't show up, and finally the lady of the house came out and said, "What are you doing?" And so I had it was a windy day like today and here, and this little bird, it's springtime, the bird had fallen out of a nest because of the wind, fell and broke his wing, and I was trying to do a surgical work on his wing. I don't think I did a great job, but I tried, and so from then on, they named me, I was 2 years old, and they named me Doc, so I became known as Doc from that time on. I had no idea what I was doing.
So it was your destiny to become a doctor, to become Dr.
No,
Valentine.
I don't know about that. But anyway, so then my next memories are really going to kindergarten for 2 years, because my teacher was a close friend of my mother's and sort of told her that she could take me in to sort of babysit me, all day long. And I went behind the horse. My brother and sisters had a beautiful painted horse, and very friendly, very very nice horse. And they rode, the two of them rode to the school, down a hill to the school. And my job at school was I had to do something. Everybody worked. All 8 grades were in the school. Dighton High, it's called Dighton High School. One teacher, eight classes, and my job was to pump the pump at 10 o'clock when the kids went out, and then again in the afternoon, I rushed out, started pumping, and they all had their tin cups.
This was the water pump.
The water pump. So I was really proud that I had a job to do, but I didn't have any classes. I just listened to all of this. I learned a lot of things from the older boys.
I'm sure you did.
I won't get into those stories.
Okay, so let's move forward a little bit and tell me about your time in high school, and maybe you can explain how you had gotten into college but almost didn't graduate high school.
Well, all through grade school, I mentioned that the teachers were all outstanding, every single one of them, and we were close to the university. That Monticello is located about 20 miles from the University of Illinois. And they were that time they were a very powerful school. So we benefited from the spin out of teachers that came. But so in high school, I had one of those fresh teachers out of U of I. I just thought all these farm boys and girls were going to go to college, and so she just assumed that. And so she told us, gave us preparation. Very few did, of course, but. She was a very impressive teacher, and so I was a good student until about the 3rd year when I had to work full time I had to pay for all my own clothes
and you were working in a dairy at the time.
I was working in a dairy at the time, and left at 1:30 in the morning to start to work, so my grades went plummeting at that point, but. We still had fun, and my buddies all had horses, and I had trained a horse, and the horse trained me, I think. He was a nice horse, but he wasn't really very well trained. He went where the food was. And so we were last week of school, we took the horses and played cowboys and tied them outdoors. That was okay. The principal was not too happy, but wouldn't tell us no. And so about the last few days of school, I was bringing my horse, we all were showing off outside the school, when one of my friends were just being nasty, they opened the big doors of the hallway to the school, which was huge. I mean, it was probably 100 yards or so long, all marble floors, and so really slick. And the chancellor's office was right at the very end, so my horse went through that door. I ducked under and about cracked my head on the ceiling, but that horse was headed for food, for the barn. He got about 2/3 down there, and I was worried he might do his thing right there on the marble floor. But I turned him about that point, and just about got out the door when the principal stuck his head out. That was too much. He kicked me out of school. And so fortunately for me, I mean, I wasn't going to, he wouldn't let me graduate. So fortunately for me, my boss was the chairman of the school board. So I ended up finishing school.
And then a few years ago, I saw that you got the Alumnus of the Year Award from that very same school, right?
Well,
they clearly didn't know about this last-second suspension.
No, they didn't, and it was interesting because the educational system in high school was very spotty compared to, I had a few great teachers, and some of them were not as good as my grade school teacher. Every one of those left a mark, but only a few great teachers. Some of them are hanging in there, like the football coach taught science. And you can imagine we didn't learn a lot about atoms, right? So I was not really prepared for the big time, but university was 20 miles away, but the tuition in that time was still pretty bad for a semester. now it was $32.50 a semester. And so, that was a lot of money to come up with. And so I had finally saved enough money to get one semester under me. So I went to U of I, and the competition was unbelievable. They had 40,000 students. You'd be in a class, and these people were really, really good. I was behind time.
And you had a scholarship, right, from your job working in the dairy?
Well,
to
not
study
a
dairy
scholarship, but the boss who'd saved me a couple of times already told me he was a U of I graduate in the dairy science field, and he wanted me to go become a dairy scientist. And I thought I might do that, and he paid for that first semester and was willing to put me through, pay all the schooling if I became a dairy scientist. But one of the jobs I had when the semi-boss was away, you had to grade the milk that came into the dairy, and to do that you need a microscope. And my job, unprepared and untrained, was to take over for a few weeks when the boss of the dairy, not the owner, but the boss of the dairy, he had to grade the milk, and your milk bill, how much you paid the farmer, depended a lot on how many bugs, streptococcus were in your milk. So you had to make a certain quantitative analysis, put it under the microscope, and give them a rating. Well, these farmers were really, some of them were really meticulous. They'd been tops of the tops, and that was it. So in my 2 weeks, I downgraded all the best ones, and it turns out that the reason was that somewhere in there I didn't clean my lens, and so a number of I was counting the number plus 100, and that really put them below the limit. And it saved the dairy a lot of money, but the farmers were up in arms, and they went. And so he quietly sort of, I don't know how he settled that, but I wasn't assigned anymore to the microscope for sure. But
so you went into microbiology then?
Well, that was my introduction. I kind of liked those little bugs. I wasn't, when I went to college, it was just nip and tuck because I needed money and it was poor and I had to stay at home. It was nip and tuck. And
so you got a job.
I needed a job, and my professor was a young professor there had just come to the University. He had an opening for a dishwasher. And
this was Ralph Wolf.
Ralph Wolf, who later became a very famous microbiologist. He and Woese, the fathers of the third
domain of life.
The third domain of life. They discovered that there was another domain of life out there. It was quite an accomplishment. That was many years later. So I started out washing dishes, but they were sort of slick, and every once in a while, and I dropped his best beaker. Well, we were on a meager. We didn't have grants. There were no NSF grants, NIH grants. If you wanted to do a cold centrifugation, you took a Serval tabletop centrifuge and put it into an old refrigerator and threw in dry ice and turned it on and closed the door and let it spin whatever you're doing. So it was a whole new world, and so you learn we calibrated our own pipette, so you're essentially using one petri dish and one pipette. Mine was a little crooked, but it was more or less accurate. That's how you did science. So it was obvious, no big time, 50 people on a paper like there is today. You had to do a little thinking, and I realized I would just I was treading water. So very early on I decided, well, how do you crack this system? Is there a code that I should know about here? And so finally I realized that, well, I read a couple of papers and took some classes on the unity principle of biochemistry. I thought, well, that's kind of interesting. And the man that wrote this particular paper said he that what goes on in a E. coli is going to go on in an elephant. So I thought, wow, that's kind of interesting information. And so then I had never been to a seminar in my life until one of my buddies said, well, there's a famous man coming to U of I from Pasteur Institute. And I said, what's his name? He said, Jacques Monod. Jacques Monod. I said, who's that? Well, he said, why don't you come? It's a seminar. It's free. You know, you just sit in the back. And so Jacques, he had been in resistance in France during the war, and then got caught and blown up pretty bad by the German. And he had his leg was permanently out. So he walked on up to that. Was giving a seminar with a cane. He was only 40 years old, 40 or whatever. He wasn't an old man. He walked up to the cane like that, and he drew E. coli on the left side of the board, and darn, a bunch of arrows, and he drew an elephant. And so I said, man, this is maybe there's something to this. And so I was influenced at that point and started to seek out all the people that thought that way. It's kind of a religious thing. And it turns out there were strains of scientists at that time, really famous ones. Some were here in California. In fact, it was sort of the starting place for that, bringing European unity. The man who described the unity of biochemistry was, in fact, a Dutchman from Delft, University of Delft in Holland. And so I found him. His name was Albert Jan Cliver. And so, gee, seemed more and more. Then I heard about this strange group of geneticists, and happened to be one of them as one of our teachers at the university. His name was Salvador Lurie. He was an applied mathematician, MD from Italy. He buddied around all summer with these people called Hershey and the whole early group. Monod would come over, and all the European Pasteur people would come over, and Max Delbruck was the dean of this, and he was the postdoc for Niels Bohr, and the greatest reductionist of all time, and second only to Einstein. And Einstein was a universalist, and Bohr was a reductionist. So this thing was spreading.
So you were exposed to this, but let's talk a little bit about your, the science that you were conducting as you start thinking about this. So you made a fairly significant discovery during your undergraduate years at the U of I. So maybe you can tell us.
It was all related to it was right out of this first chapter of Unity Principle of Biochemistry. In 1929, Cliver was an organic chemist, hybridized to do microbiology. So the hybrid vigor idea was, and it came to America through Stanford's appointment of Cornelius Van Niel down there. He was Cliver's student, so he was sort of the emissary for that era. There was no molecular biology. At that point it was going on, but nobody paid any attention to it. They didn't think you could do E. coli and bacterial viruses and get anywhere in terms of evolution of a tree, of a redwood. So they weren't too happy with that group. But now Cliver was a different story. The biochemists picked up on that pretty quickly. And Cliver had decided, he made a phenomenal discovery, and published it in 1929, I think it was in the JBC, one of the major journals of the era, that the Swiss had a small country, and they developed all the early colored dyes for making green and blue and whatever, and the chemists became experts. They were the world's experts on dyes in chemistry. They had a bunch of strange chemicals that seemed to be, under one condition, they become brilliantly colored, and then shake them in air, and they became colorless instantly. Yeah. And these are called the Viologen series. They became herbicides later on, but they also became, nowadays, they're the main way to commit suicide in developing countries, because
So you capitalized on these. Why don't you tell us about what you did?
So my job, after I broke all the glasses, after about the week, he said, we're going to promote you, and why don't you just work with this finishing graduate student? He was a very academic man, very nice. Let me grow his bugs and make the extracts and fractionate the extracts and go as far as I wanted to go. I was a sophomore or junior when all this came together, and I got a hold of Cliver's paper, and he'd already taken those dyes, and they were free radical dyes. They throw them in with a liver extract or an E. coli or whatever, and they would become colored sometimes. And at that time, they just developed the redox potential at Berkeley, that you could score oxidation reduction. Physicists already had this back, and they had it backwards. We're now backwards but anyway, it stayed backwards forever. And Cliver had found that the strongest high-energy electron in biology was around 0.7 volts minus 0.7 volts. Now with a billion volt, cyclotrons and everything, that doesn't sound like much, but that's pretty powerful. And then on the other scale, oxygen is, but he didn't deal with the low part scale. We found, plant scientists found that one. It's called the photosystem 2 reaction.
Yeah. So but let's talk about the system that you developed and what you did with that dye system.
It was a very simple experiment. We took all this together, go in the cold room, roll up your sleeves, go grow the bugs, make the extracts, and do everything you can think of in the world to see the extract. You'd put in pyruvic acid, and out would come CO2 and hydrogen. So I was interested in the hydrogen because it's clearly a high-energy electron. How did it make the hydrogen? And Cliver's dyes were one way to check this. I finally made an extract that would not make hydrogen anymore. We had all kinds of tests. Fritz Lippmann and my other great...
So it was missing the key electron carrier.
Missing this key electron carrier. And it was clearly a high-energy electron carrier, and it was donating to make H2 come bubbling out of the sample. And so we knew the electrons on pyruvate were hot. They're minus 0.52. And that was minus 42, but still, that's high energy level. No one had ever busted that apart.
Yeah.
And photosynthesis, photosystem 1, and photosystem 2 make high energy electrons.
So your advisors helped you to get a summer position.
Well, once I found that the viologens would replace this, I also had fractionated what's left in the fractions. If I split the fraction into the main protein components by running it through a particular kind of column, I got an extract that you needed to put methylviologen in order to get hydrogen. But I didn't stop there. I took the remaining stuff that came through and started to try to fractionate it to see if I could purify against that assay. And that was the crucial breakthrough that was needed. That was really what you needed. So I'd already fractionated that and found out there's something weird about this brown mixture. And I thought, gee, this is weird. And I didn't have the background in chemistry to sort of realize what might be going on. So when my advisors got together and heard the story, we published this in JBC. Oh God,
mid-50s sometime.
Mid-50s. But it was just a little short JBC story that tells this assay. Nobody ever took a look at that. But they told me, you got to get, I went to see one of the people in my Wolf's third generation. It was Gonzalez, O'Shane, Wolf, and they all sort of did things the same way. But Gonzalez was the head of biochemistry at Illinois, and he was instrumental in bringing Wolf there. Yeah. And so he heard my story, and he was notorious for just dropping a conversation and just going to do something else. He had a real temper too. So I went and told him the story, and he didn't seem to be too interested. So I waited and waited. He seemed to be calling people some other unrelated thing. I left and started to go back to my lab, and I'd gotten all the way into the hall when Gunny, he did this routinely apparently. He yelled some curse and said, "Get back here. You're going to arrive in DuPont experiment station June 11th. And I said, "What?" He said, "You're going to join the experiment station." And they had just cracked the code for nitrogen fixing. They needed high energy electrons. Well, they didn't know that. They knew that they could take the same extracts I was working with and drive the nitrogen fixation reaction. Had the whole reaction, but they didn't know how to break it down. So immediately he saw the probability that this was related. So they sent me out there for the summer, and in 11 days, I'd fractionated it to purity, had 12 grams of the dry material, and started to take it to analytical groups. And the first thing they said, "You've contaminated this mixture because it's solid iron. You've got rust in there, you've got nails in there or something." And they said, "Well, no, it works in this assay." So
you took at DuPont, you took the mixtures, the various fractions, and then compared them against the assay.
Oh,
that you had developed?
We were big time there. I didn't have to make bugs anymore. They had 4 people in a room, half as big as this building, just making the extracts of these nitrogen-fixing organisms. And when we did a prep, they had industrial kitchen-type 200-gallon stainless tubes, and use the canoe paddle to stir your mixture. And so one of those was enough to get 12 grams of pure. I put it in 12 bottles, and you could freeze them. It was kind of like a glass. It was a glassy sort of brown. But in the meantime, somebody told me, "Well, you got to take it analytical, get other elements." So we took it over to the group that did sulfur. And they said, "Don't come back." We heated this stuff up, and everybody left the room. So you made rotten eggs. It's rotten eggs. It's
rusty rotten eggs.
Nails. This can't be. But then finally, I said, "It's working. I don't care what you think it is. It's working." And it was easy to fractionate. It was like one-step procedure because the molecule turned out to be very acidic. And man, you get and the latest, they just discovered how to fractionate proteins through various columns with different charge groups. You put a ton of this stuff on there, and it stuck like you couldn't move it from the top until you put a half molar or whatever gradient of salt. It would come out pure.
So you've now discovered this very high energy electron carrier.
Yes.
Tell us how did that get named?
Well,
what is it named?
I got half the name. We had a Ferry something, and so we didn't have the dachshund, but they had a contest. We couldn't agree on this, so they held a contest at DuPont. We didn't know the universality of this thing. I didn't either. I just had it there, it's working. And so Ferry something, and it's finally somebody came up with the Greek word dachshund, whatever, it just fell into place. But the winner of they gave me the winner because I got the Ferry part. The person, I forgot who did. I didn't do the Ferry. And a huge reward because it was a 25, in those days you could buy a Baby Ruth candy bar about that big, a foot long, and you'd take 3 or 4 days to eat it. It was a monster for 25 cents. So that was a reward for naming Ferry dachshund.
For discovering and naming Ferry dachshund.
The thing that was most interesting about this for agriculture, which nobody knows these stories, of course. This is the first time this has been gotten into the public domain, is that we had as a consultant at DuPont, Gunny Gonzalez was the main, he was the one that got me there, but he didn't know electron transport that well. As a group at Cornell, Bill will know this guy, the two most famous, one of them was Ephraim Racker. He was one of the world's experts on respiration, electron transport, and the other man was his partner up at Cornell that did the chloroplast side of this world, and they had all the fractionation. So I had decided that I put on a gram of this stuff. I put on there Cornell. I knew he was coming in a week. The consultant, not the chloroplast guy, but Racker. He was a famous guy, and very nice to talk to, etc. So I was going to slip it into his pocket and have him tested in the chloroplast tests. And our boss of this particular group was an organic chemist given the job because he'd made some nice piece of chemistry for him. He didn't know biology from anything. And so I thought, well, I'll be honest this time, and I better ask him. So I asked him, and he said, no way, we're patenting this. I said, what do you mean you're patenting it? Well, you know, we're going to patent high energy. You can't do that, actually. But he thought you could. So I couldn't just, if I did this, I'm in trouble. So I decided, okay, I'll wait. Well, and then the word got to Dan Arnon through 6 months later and we'd published the first papers by that time. But one of the people at DuPont, desperately wanted to go somewhere else, was not really part of the program, but he got involved in some of the chemical parts of it later. Actually came out here 6 months later and gave a seminar in Arnon's group and told him the whole story before it was published. Well, if you know Dan Arnon, you're going to understand what happened next. He discovered ferredoxin for photosynthesis, and that was the biggest boo-boo I ever made in my life. And being honest, in that case, was a big mistake because it was absolutely the same thing. And San Pietro had already named his molecules the San Pietro factor, but he missed it because it was San Pietro factors, plural. And we had purified it. It was two enzymes, one that connects, one is a reductase that ferredoxin reduces, and it's the reduced pyridine nucleotide reductase. So that was basically the way it happened, and this was buried because DuPont, then they asked me to stay, permanent employee, and I didn't want to, and they got mad at me and got me moved down on the list of people who came up with the assays. It was all they don't know to this day.
And so Ralph Wolf has covered a little bit of this also.
Well, he covered that.
He
covered it pretty well. He covered that interesting part of the story, but not the whole story.
So let's move forward. Otherwise, we're never going to get to hear about your career at UC Davis. So you finished your PhD at the University of Illinois with Ralph, working up additional aspects of the ferredoxin story in bacteria, and then you went on to postdoc. And why don't you just tell us a little bit about what you did for your postdoc with Zinder at Rockefeller?
Well, I don't know where I got influence to do this, but remember when I first took any genetics course, they were given by Sal Luria. Well, by this time he'd moved to MIT and was about to win the Nobel Prize. So the three of them that he buddied around with all won the Nobel Prize about that time. Delbruck, Luria, and Hershey, they were given it for being the fathers of molecular biology. And so I said, maybe I'd done genetic engineering experiments along the way in probably late 50s, early 60s. I started doing genetic. I was so interested in an experiment that Luria's lab reported. He and Ed Lennox, Ed was an experiment there on sabbatical, famous guy in his own right, but not like Luria. They had discovered a way to move genes. Norton Zinder, who I did my postdoc with, had discovered and wrote his thesis up in Scientific American. It was a big journal in those days. It's called Infectious Heredity. And he discovered viruses could pick up genes and specific genes and donate them to a new host, and you got that gene would operate there. But what Lurie and Lennox did, they found a virus that was a pretty good sized head, would package about 11,000 bases. That's enough for a whole operon, 10 average genes. And that, you had no viral genes in there. It just had the package. So if you grew this organism with this virus, a certain percentage would represent all the pieces of the whole chromosome. So if you had a selective system for nitrogen fixation or whatever, this virus is duck soup. And so I didn't believe it. So I asked Lurie, and we weren't getting along that well, but I'm going to get into the reason. I asked him if I could get some virus and reproduce that experiment. Of course, he was happy to give me the culture. It was very simple, the culture. And so I repeated that in class to demonstrate to my professor I was TAing in a class called bacterial pathogenicity. And this man was trained as a vitalist. And he still believed in vitalism. I don't know what vitalism was. Well, that was no self-respecting living organism would be alive if it had to depend on anything else outside that it can't make. That's vitalism. I said, that is crazy. So I got in trouble with my professor big time, and he let me lecture each time a little bit. So to prove him wrong, I took his favorite selective system between Shigella dysentery and E. coli. And E. coli is plus for this gene, and this one is minus, and so I'm going to take, this would be illegal now, but take the lac gene from there and put it over there and totally mess up the selective system for forever, which was the standard. This is the standard central dogma of how you... Yeah. And so I did the experiment in class, and it worked beautifully. But my professor still believed in vitalism. He said, you contaminated the system. But I knew I had.
And this is the early 1960s when this happened.
Probably late 50s
late
or
50s,
60s.
Early 60s.
So that was my first real genetic. So I messed around with genetic engineering. We won't get into the background, but I had some really wild stories that started to go in various directions. But I never really followed up on it, and none of those experiments worked. Yeah. That went way too far. You should stick with one gene. But that was the background that led to the single gene hypothesis here at Davis that later I thought about in the
1970s probably. But let's before we get there. So you were, let's just follow your career briefly, because you were at Rockefeller, and then you started, and I think you went around and did a couple of stays at other places. But then you were offered a position here at Davis in the early 60s, but
yes.
you instead went to Berkeley at that time.
Well, I'm going to go fairly quickly through this because it's all very interesting, but we'll never get to the end of this if we keep on. We got the background, but basically this ferredoxin was a pretty big deal, but I could have stayed with it, and a lot of people, I got plenty of job offers from schools.
Well, we're still working on it today in different ways.
They wanted. I had no idea that was going to be so critical around. We didn't even see. I saw the chloroplast, but I didn't see the mitochondria. That was the system is pure iron all the way through there, and complex 1. It's got 7 of them in a row, a wire, 2 ferredoxins, but had no idea. So, but somehow Lurie convinced me that you ought to know some genetics. So I went and asked people at Stanford. I tried to get into Kornberg or somebody's lab, and I didn't make or Berg's lab or whatever, but I didn't make it. But they didn't know. They knew a little, but it wasn't very genetic. They wanted somebody already trained. But Rockefeller, there was a famous lab there. It's called the Avery Lab, which was run by Ronald Hotchkiss, and he was the head of it. And Norton was his senior person, and so it was the Avery chair was Hotchkiss, and then, but the most famous was Zender, because he'd discovered specialized transduction, and he was a student and finished his thesis at age 18. PhD discovered that, and his boss was a man named Joshua Lederberg, who won his Nobel Prize for sexuality and bacteria. That's where the whole story of how that happened. So we were tied in, I was tied into that group. I knew all, they were all tied, they were sort of a separate entity from the Luria Delbruck group, and this was, Lederberg was so powerful that he was, you know, his own sphere of influence. And but Zender was a tough guy to get along with. Everybody knew that, but he was a brilliant guy, and he discovered the world's smallest bacterial virus, and he thought it was only one gene, but it turned out to be 3. So I said, well, that's a good place to start. So Dan Nathans had published a PNAS paper where he had extracted, you could grow tons of this virus in big vats, and it turned out to be RNA. And so it was messenger RNA. So here you are, you've got now a polycysonic message. Now TMV was leading the world.
Tobacco mosaic virus.
Tobacco mosaic virus leading the world, but but it didn't have this, you know, the utility in terms of speed. You get an answer, you do it in the morning, but before you go home, you had an answer, and at least figuring out what was going on with you. So I went there to follow Nathan's paper. He was with Lippmann as a postdoc, and I got to know Lippmann extremely well. He was a really brilliant guy, and Nathan's later was pretty brilliant too. Both of them won Nobel Prizes. Nathan's won it for recombinant DNA about 5 years later. So these were not, you know, lightweight guys, and so but I never really progressed very much. We published a lot of papers in viruses, but no big slam dunkers. But I learned a lot about philosophy and in Avery Lab. That's where DNA name was named DNA, and of course the poor guy was persecuted beyond belief, for he died. Still, people saying it can't be DNA. That's a reserve for phosphate and nitrogen, and it's got to be proteins, right? I mean, this was incredible. He never won the Nobel Prize for his 42 paper, but believe you, if you went there and listened to Ron Hotchkiss, you had to take a week of learn Avery.
Yeah.
And so he took you through the whole history of the Avery discovery.
So you started working on viruses, and you learned your genetic engineering at this point, and then that took you to a faculty position at Berkeley. Do you want to tell us just quickly what you worked on there while you were at Berkeley?
Well, actually, I was interviewed, I think, at 5 places, and the University of Washington. I basically could go to any of those places. It was my paradoxical work. It was a stepping stone, but this area that I'm talking about here had potential, but I was not really. I'm more of a bioenergeticist than I'm a biologist. But anyway, I was offered. I came out to interview at Davis, and Paul Stump and Kahn just died, I understand. They were a pretty powerful group here. In fact, plants were king out here at that point. And they were sort of the top of the top. And so I gave them a talk, forgot what it was exactly, but it was viruses, not ferredoxin. And it was a hot day, and I got on the way to the airplane. I thought, well, you know, I'll get a note from them a month from now, tell me how I did. They offered me a job on the tarmac. Hot tarmac. I thought, oh, wait a minute here. I don't know what to do. It was a new building. It was very, this is a real opportunity. And then so I didn't have time to think about it. And I'd been told by peers at Illinois, be careful about where you end up. And so, there was the distinction between Berkeley and Davis at that point was really different approaches. Yeah. So, but anyway, I thought it was a great opportunity, but I wanted time to think about it. But I didn't know the etiquette of politics, you got to be very delicate with this stuff. So in the meantime, they paid my way out here, Davis did. But I knew Barker I'd spent some months a summer with H.A. Barker at Berkeley, who was chair of biochemistry up there, and we get along great. Actually, I did my thesis on a project, not on ferredoxin, but on a project that Barker let me work on for months, and I made my PhD thesis out of it. We published that in JBC. It's a very interesting story. So, but they were not meetings. They had an opening position, and he was interested in me and wanted me to apply, but they couldn't make it. The committees are not going to meet. The machine stopped until people got back from summer vacations. That's a couple of months away. So, it was that 2 months. In the meantime, he sort of let me know that they're quite serious. So then later on, I went out there on the same trip and gave my talk. And it was up
on Davis's dime.
Well, it could have. I mean, I didn't even know about who's paying for what.
Yeah, so you interviewed at Berkeley.
I broke about 3 rules here, 3 strikes already, not knowing how to handle this. I like the lab. I don't do this politics. So, Jesus, everybody showed up at the virus lab to hear this story because this virus thing, I mean, they were the kings of TMV, the Nobel Prize. The building was named after the guy that discovered you crystallize the virus, and his proteges were there and close to cracking the polycitronic message, and so they were obviously a little bit interested. So this is a powerhouse group of people that came. The place was packed, and my talk was mediocre, but just the idea that you could suddenly all of a sudden blow out TMB. I mean, he didn't say that, but I didn't even know that. But this was 10 times easier to prove.
So this was what, 1963-ish, 64? That's okay. But
63, I think.
So they offered you a job, and you took it.
They offered me a job, and they took it. They had a new building coming up, and so I took it.
And can you say, I know that while you were there, you did quite a bit with nitrogen fixation, and that really got you into the nitrogen fixation world. Can you just tell us briefly what that critical experiment was that led you to to
there's 2 parts
genes?
of this. There's an A part and a B part, because I really wanted to still apply molecular genetics to agriculture. I'm an ag background, and so I wanted to apply molecular biology to agriculture somehow, and I had no idea how to do it. Then I decided one day, well, open the book, Delbruck, chapter 1, and how would you do that? And so years ago, I thought about this for years. We had no idea what nitrogen fixation was, what the biochemistry was, what metals were needed, nothing. There was no genetic, 0. And the reason is the bugs that were popular for the biochemists were the worst possible organisms to do genetics with. So I was reading this article on the nitrogen-fixing people of New Guinea, and they were doing nitrogen balances on these people, I guess species and and they had positive. There's more nitrogen coming out than went in, and so this led to some groups that isolated the bugs, and they're all Klebsiella. Well, I knew from my experience with Zinder all about the evolution of the enterics, and I knew P1 would transfer. So here you had a ready-made. If you get
that was virus P1.
Virus P1
would transfer the genes to Klebsiella.
It was, but so I wrote Wilson at Wisconsin, he had a collection, and 3 or 4 people had collected these. And it was quite a deal if you could get nitrogen-fixing people right. That'd be but it turns out that, and the National Science Foundation really interested in this. So the people who built my first house in Davis were postdocs in nutrition. They built it as a spec home, and they were awarded the first NSF grant to go to New Guinea. No,
Papua New Guinea, is that what you said?
one of there's a big island over there. Anyway, it was a protectorate of Australia, so to get permission, you had to get permission from the Australian government. The NSF gave him the grant, and the experiment was you got to put the person in a bag, and they'd already discovered a neat way to measure nitrogen fixation. You close it off momentarily, leave enough oxygen in there, right? And then you shoot welding gas in there and wait a few minutes, and
acetylene reduction assay.
This hot electrons, man, it takes about unbelievable energy punch to get this thing broken. You need 14 ATPs, an umpteen number of high energy electrons just to smack that triple bond open. Nobody had ever run into anything like this. And so, but it was easy to measure it by this new assay. And so they'd gotten permission, money from NSF, selling all their stuff here, and we bought the house that they built, but they weren't living in it.
I do wish, by the way, they'd put the windows in the proper direction and it didn't leak, but go ahead.
So anyway, that experiment got me to Klebsiella, and I'd already had the background to realize that if you got it in Klebsiella, you had P1 transduction. So but nobody cared about the biochemistry in this book. They were established National Academy of Science labs around the world, and this is how you do nitrogen fixation. So I was a maverick, and I didn't realize the politics of being in that particular position, because the old-timers did not like this story. So out of the blue, unconnected but here's where I made a lot of mistakes I made in my career. This is one of the big ones. I should have gone in there, but so I just did it totally independently using a new kind of philosophy. Well, you should have tied, as a plane a few hours away to go to Wisconsin and work with these people. And they'd offered me a job earlier to do this at Wisconsin. And it's very close to taking that job. But, well, anyway, that's another story. But in the meantime, they'd hired my colleague from Wolf's lab, who was following along, watching this story. He'd helped me a bit on ferredoxin. And he was offered the job at Wisconsin, so he was there. Winston Brill, he got the academy because of his discovery of the nitrogen fixation gene, right? So these stories get a little bit, typical.
So you were working in Klebsiella, and you worked out the genes for nitrogen fixation in.
Well, if you look at this paper, it's a really simple paper. It went in about a year before it got published. It was just simplicity. It was an assay that you saw. You could tell. You didn't need any machines for this. You used your eye. It was the nitrogen-fixing colonies had to be put in a certain kind of anaerobic medium, which I learned how to do this stuff from Wolf. It's rather tricky. Not as bad as methanogens, but pretty bad. You don't want any oxygen, and it cannot fix nitrogen and oxygen. It has to be strictly anaerobic. So they're brown. They make so much ferredoxin-like molecules, the whole colony turns brown. When you make a NIF- mutant, it's white or creamy. You can't miss it. So we set up an assay, and easily made 100 of them, whatever they were. But that required an anaerobic. So that it's all given in that PNAS paper is how all this happened.
Yeah.
But the key thing about the paper, we then could use the P1 transfer system to show you could turn a white to a brown, And then,
so you could reintroduce the gene that had been
or the
lost for
genes,
nitrogen fixation.
and so you could show that led to the naming of the genes, and the technical name according to the book, the name should be nitrogen or dinitrogen assimilation or nut, N-U-T, and so
Didn't want the nut gene.
I didn't want the nut genes. I thought I'd better do the NIF gene. So the paper describes all the background that you needed to bust this problem wide open.
Yeah, and I've looked at that paper recently, and I note that the naming of the NIF genes in that paper is actually a footnote, that using a convention that you named them the NIF genes for nitrogen fixation.
Yeah,
Which I think most people don't know. So let's move on from Berkeley. You ran into a few problems there, but ultimately you were offered tenure but declined it. We're going to get to Davis, so I want to skip over some of those.
That is true.
Some of those biopolitics and move on to San Diego.
When the NIF genes hit the scene, then Dr. Barker was so pretty influential, and several people realized there's some possibilities here and so they offered me that. They had that new lab upstairs, a brilliant lab. But the man who was going to get that lab, the heir apparent to that lab, had been murdered by one of his students a couple of years before, so it never got. It was a tragic story. He was a plant biochemist.
So they offered you the lab, but you instead opted to leave and took a 5-year, as I understand, career development award granted by the NIH that allowed you to travel all over. And I don't think we have time to touch on each of your
No, we will not do that. That's
travels.
a story in itself.
But you ended up auspiciously at UC San Diego for some time, where yours truly came to be. And then why don't you tell us about the transition then to coming to Davis when you truly joined the faculty here. So that was 1974, I believe.
Yes.
And maybe you can tell us a little bit about how that came to be. How did you end up at UC Davis?
Well, there are lots that would take a month of discussions, but basically I had a 5-year sabbatical and good pay, $50,000 a year for little cursory expenses in the lab, and guaranteed 5 years. And anywhere you want to go for 5. So you go to Paris and La Jolla. Went to La Jolla, right? I was about ready to dance around the world for 5 years. They stipulated in the award that you can't do much teaching. So this right away really made all the senior. Here's this guy coming in, doesn't have to teach. So that's why this program soon after was
it was changed.
It was changed.
By the time I got it, we had to teach.
For 5 years. So this was an incredible opportunity. So I was into that partially when the idea of a plant growth lab, which was integrating modern molecular genetics, biochemistry, plant physiology all together, and up here at Davis, the post-harvest group was famous at that point for they did just that, and they're still top notch. But I mean, that group made big discoveries in post-harvest physiology, and in fact, I think ethylene, the whole story of ripening. I mean, I don't know the history. But it was a big success. To me, it was a big success. It wasn't. So this was supposed to duplicate that, and they used a different format rather than a department to use these. Well, that was a success story at the time.
Yeah, I think we call them now organized research units on campus.
Organized research units.
Yeah, and so that was late 1974, 75. And it always seemed a little funny to me that you joined the plant growth lab, because in my experience you never worked on plants. But I think, as we've heard, you were looking at the potential of molecular farming. Can you tell us a little bit about how that term came to be? I've heard stories of you on stage with a pitchfork yelling, "I am a molecular farmer."
Well, that's probably true, but.
It is. Gerhard Gottschalk pulled me aside at a conference and told me this
he
once.
was at a conference.
And he remembered it 20-something years later. So how did you become a molecular farmer?
Well, the term was probably floating around already, but I didn't really historically look at that. But I defined my own molecular farming, and the real first official use of it in front of a big audience of very prestigious people was after the group at Washington. They decided that they didn't want the molecular biologists working in this genetic engineering field didn't want to have the reputation that the physicists at that point had when they had this big, and today 55%, well, look at the president, he doesn't believe in science, and 55 or 60% of people don't believe science is useful. So that's your starting point. And so in order to avoid later, they decided to have a group of people got together and had an international called an international moratorium on doing any experiments in genetic engineering until it was sorted out as the benefits and risks. And so it was a two-part system. Actually, it turned out to be a three-part system. First, the politicians of Washington, D.C., and all the center of our where all your tax money goes, go there. The first one was there, and it's more. This is basically, if you look at history, it's mainly been a medical story, not ag story. But Paul Berg and others who were, and he newly Nobel Prize winner, the people he chose to work with him and form this were very prestigious people. They still are. Some of them are not alive, but they really walked the chalk and narrow line and knew that they needed a meeting, and it couldn't be immediately evident and biased towards positive benefits. They had to give both benefits and risk. The meeting in Washington was a trial balloon, but with at the same time all open forum for any assistance of the senators or whatever could come to this meeting. And so very last minute, Paul Berg called me to ask me to give a seminar on this nitrogen fixation story. So I got up there. I'll show later my.
You were asked to appear on one of the morning shows, as I understand.
Well, Wake Up Washington. We'll get back to that story later. But I brought my memento I still got it. It's faded a bit, but it was really brilliantly. It cost me about $17 to put that picture on there, but I got that picture from out here in
Baby Ruth's.
From Davis's.
So you gave a talk there, but then you were also involved very much in this genetic engineering debate, and you were asked to be on this Wake Up Washington. So why don't you maybe tell us a little bit about that?
Well, Wake Up Washington, I reluctantly did this for reasons I should not have ever gotten into this, but I decided to do it, and I was debating a really sharp professor who had a sharp tongue from MIT, Ethan Signer, and he was a smooth cookie and very good scientist, but he was against, and I was the pro, and I tend to get tied up when I get in front of an audience. You never know what I'm going to say, so it went just as I expected, that he had every argument in the world. I mean, I was snowed under before I got started. So I knew this was going to happen, so I thought I need some sort of a shock treatment here, sort of like, our president uses these shock treatments. I thought I better have something. And I think they're going to take a picture and record what my shot was. And so at the end of this, it was, I was wiped out completely. I mean, I couldn't defend anything. And so I had, under my shirt, I had a T-shirt, and I had on the front of it, I've got a beautiful shot of the early soybean variety here from Davis, out in the field. They grew up the seeds occasionally, and the original soybean was this big bush. It went out like that, like a small tree. And we didn't, in the 30s, when they first started growing it, they didn't know what to do with it. And so they baled it and fed it to cows, and cows hated it. But if you're starving, it's got it's got plenty of nitrogen in it. So they had the seeds for those early varieties, and so I went out one time and found one, pulled it. It was pretty moth-eaten on the top, but it was a big monster. So I pulled it up, and the nodules were incredible. They were absolutely incredible. So I had that emblazoned on a T-shirt on the front. It's called solar. Well, I don't know what solar protein or whatever. You'll
solar
see
protein, I think it was.
You'll see solar protein, and so in the back said,
something about fixing
sustainability or something. Anyway, I thought it was so. I'd planned to, without telling the guys doing the various, and this was in a room of beautiful. The palm trees leaves were there in front of you, and you felt, big, easy chairs, and in and out with all the refreshments you needed. This is high-class stuff. And you felt, felt at ease. And so I felt pretty much at ease, so I didn't think too much about it. So I opened without telling, then the camera was off of me for a while, so I opened my buttons, and so I was going to just lift the
upper shirt.
The upper shirt off. And when it did that, I forgot about static electricity. And so the camera was looking at me right down there, and it was focused exactly on my belly button. And, I'm not that hairy, but it was not the best-looking button to wake up to in Washington that you could see. And the guy, he was so flabbergasted, he should have pulled it off of there, but he kept that thing going for, seemed like eternity, going out at 7 o'clock in the morning to all the people watching television.
So you've gotten in trouble a couple of times with soybean root nodules. So as you've told me before, your teaching responsibilities at Davis really were focused on doing lectures for visiting dignitaries or groups that were coming to campus, and that you were very much a designated lecturer for the dean's office. 1977, Prince Charles came to visit.
Yes.
Why don't you tell us how you used soybeans to your advantage there?
That was my final blow. It's like 3.5. The chancellor was supposed to be on Monday morning. Prince was coming on Saturday at the end of a 10-day trip around the United States, and he'd gone that morning, Saturday morning. They'd picked him up by helicopter, took him over to the Napa Valley to see wineries and maybe do a little tasting or whatever. And then they had this helicopter fixed up so that there was sort of a semi-door, and they wanted to show him the agricultural. If you think about it, you'll see grapes and a helicopter going pretty fast. You can see the gradations of agriculture, and alfalfa, and then tree crops, and agronomy. It's all in rice and all at the same time. So they wanted to see when I talked to him later. So the dean had arranged for the Davis campus to give him a quick seminar, and there were 3 people, I think, chosen. I've forgotten. Well-known people.
You were one of them.
I was one of them. And I think I might have been the first one. I don't know. But the chancellor was so intense about the story that when we had practices about every other day for a week. And one day he came in, he had a screwdriver in his pocket. What the heck? So I watched and looked at him. He went in, the boys, we were going to do this in the Wixon Hall, up that stairs, a nice stairway, and but Prince might need to go to the bathroom, right? So he went in the boys' room. It stunk a little bit. I could agree to that. And then he went into the girls' room and came back out, and he took his screwdriver and he then took the screws out of the boy and switched them. So
Brilliant.
That's how serious he was.
That's what you get for you want a good chancellor. Okay, so Prince Charles comes and you give him a seminar.
Start the lecture. You're 10 minutes plus or minus 7 seconds. He clocked you, and he didn't want me to be. Charlie put me there. I don't think the chancellor was
that's
too happy.
the Dean Charlie Hess put you.
Yeah, Charlie Hess was the dean. And so here's the alignment. We're giving, nobody allowed in these rooms, no anything. And the G-men with their Thompson machine guns,
British Secret Service or something.
Our Secret Service were on top of the building with half of it, and the other half was the G-men with Thompson machine guns from Britain. And you weren't, we were given instructions. You had a booklet like a fancy dance with a little red dangly thing, and different chapters. And so the first page was Prince Charles' name. It's a whole page of, I can't even, but also you don't make your point by punching him in the hand. You do not punch it, hey, what do you think of that, So, man, we went through every detail of what you do. So one of the three had been chosen, it was me, to go separate for 20 minutes in that in the growth room over at Plant Growth Lab, where all the calluses were growing and the light was on you, and 20 minutes is all you could take in there. So that was later on. But I opened my slide, and I'd figured out what I was going to talk about. I was going to talk about super promiscuous sexuality. I thought
between plants and bacteria.
Well, I didn't say that to the prince, but I started out. That was the end of the talk, but I had to build up to this, right? I had to go through sexuality when it was discovered in 1946, and then super promiscuous sexuality in 54
between bacteria and bacteria,
and then
but you didn't,
and bacteria and plants, and was well known at this point. And so I thought that's I'd get his attention. Well, first slide, second slide, 10 seconds in, he goes to sleep. And he's lined up with the prince, the British ambassador, the the chancellor. You can see seated in those little chairs up there, you can measure that, right? But kabong, kabong, kabong. And that's the way it went. So that morning, I was getting a little worried that, I'd been through this thing down at Silmar, and I wanted something to kind of break the tensity of the tension in the room. So I went out again and got another giant big soybean, and I went into the lab early and washed those roots, all the dirt off, and washed the leaves and the bugs that did their job on the leaves, but I didn't care. It was a big plant, like about that big. And so I wrapped the nodules in white, fresh, unspoiled lab white towels, and I had it somewhere there stored. So when he went to sleep, I thought, what am I going to do? So I goes over, and I grabs that thing, and I was going to put it it's big enough to cover from his crotch. The leaves were unfortunately in the crotch of the chancellor, and I'd forgotten to take the water off of it. So when I plopped her down there, the prince sort of woke up, and I said, "What do you think of those nodules?" Well, that was a bad set of words right there. Then, by that time, the water was dripping through the chancellor's pants. Well, we won't get to that. And he was turning red already. I told you so. This is a dangerous guy to have him up here like this. So anyway, I started, what do I do? So he woke up a little bit. Then he went back to sleep again 3 times. And I just went ahead with my talk and got the super promiscuous. And I thought he was sound asleep, but he'd been drinking. 10-day trip, no sleep, wine in the morning, blah, 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 blah. Anybody's going to fall asleep. I mean, natural. So I just went ahead with the talk and went right into the super promiscuous sexuality, and I thought he didn't hear any of it. But then, like a lot of scientists that go to sleep in seminars, I know a lot of them, Delbruck went to sleep every time. he raised his hand like this and said, "Could you tell me more about super promiscuous sexuality?" And of course, the prince was asking the question, and the chancellor couldn't say, "Don't do this." So I repeated. And then we left. Oh, the other guys gave their talks, and I don't know if he went to sleep with them or not, but then I went for 20 minutes alone without anybody in there just to have a conversation with him one-to-one. And what amazed me is how small he was. He was about up to here on my shoulder. I've seen him on these big stud of horses. You'd think he's a Spartan, right? Well, he's a tiny little guy. Very easy to talk to. I mean, he's trained to all these people coming up. You got to be able to sort of interact immediately. If you're going to go, you're not going to be very happy very long. And so he's easy to talk to. And he asked, he told me, he said, I'm trained as an engineer, pilot or something. And he said, and my advisors before I came on this trip wanted me to, he was obvious, the next king. It was before the whole fiasco with Diana, but he looked like he's being readied for this job, and they wanted me to learn a little more about agriculture, because Britain is an agricultural state, just as country like we are, but people don't know that. So anyway, he said, so I particularly interested. They told me you should prepare and read a book on this topic. I said. He says, that book is called The Secret Life of Plants. And I'd read, the guys that wrote that book had written a bestseller a few years before describing how the President of the United States was 7 days in May, how he had been captured, what do you call it? The group had captured the President and took him to a location against his will.
I don't know the story.
Well, I forgot this book, but I think it was the same group, bestseller. They were feeling their cheers, so they wanted to write another bestseller, and their choice of title was terrible because it described how plants interact with music and other things that make them happy. And so he asked me, what do you think about this? And so instead of going on, I'm sorry about that one, I said, well, that's very interesting. And, that was I read that book. So anyway, that was a Prince Charles thing.
So as I understand, you made a mistake, perhaps, of talking to a reporter afterward.
Well, the reporters are the big Cadillac convertible came in, and he hopped into it, and it was a beautiful morning by then. The rain was over, October, I think, or when I was here. And they had a rope. You had to stay behind the ropes. And when he first came, the place was packed. When he left to go have a asparagus sandwich with the prince, he took a tour around the campus with the chancellor told him, make sure those polo ponies have their rear ends away from the prince, and that sheep barn, that moss or that lichen on there has been green for 27 years. Clean that up. And so off the prince went, and so everybody disappeared. Nobody but one person, way back in the back, pushing a bicycle, came in and introduced herself and said she's Paul Stump's daughter. I said, well, I got to talk to her. And she said, we can't afford to go racing over to Sacramento. And so I've been asked to stop, and would you be willing to just talk to me for a minute about what went on inside? And I said, I'm happy to do this. I said, well, the prince is charming, easy to talk to. I didn't say ordinary person, but I was trying to make him sound like a person you could, really okay. And but like anyone else, and he asked very bright questions. And like anybody else, though, after a whirlwind tour, and tended to nod off a little bit. So she wrote all this down, took it to her editor at Davis Enterprise, and Sunday morning, the next morning, the headlines from the San Francisco Chronicle were the same typeface as the
World War
surrender
II D-Day,
of Japan. About that one. Prince Charles Day in America. Day of wine and dozes. And fortunately, leading the whole front page, starting there, blah, 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 Dr. Valentine, Balentine, put him to sleep. I said, that's a typical professor who did that. And that day I was leaving, fortunately, and that afternoon I was leaving for a talk somewhere. And I had that morning, so I was walking down from coffee or something on the quad, and about half a dozen people stopped and never paid attention before and said, hey, you put him to sleep, did you? I mean, this is pretty good. But the editor said, this might be an interesting story. We better put it on Reuters or World this or that. And they said, well, just put it on all of those news things. And it went around the world. The chancellor he was supposed to be in Bombay or India on Monday mornings. He had Sunday to get ready and travel over, and Monday get his great award in nutrition in India. And he canceled, and he and the vice chancellor had to spend Monday writing a letter of apology to the British embassy, which is totally, I think, nuts. But
okay, so let's we just have a few minutes left. So there's a couple things I want to talk about. So Prince Charles obviously has come out fairly anti-genetic engineering over the years. There were other folks who were also anti-genetic engineering. I understand that once the Unabomber used your company as a return address on a bond that he sent, as just an example. But I want to go and move on to your transition at the university as you began to start up your own company. And I know that started with these meetings that you started to talk about, particularly the Asilomar meeting. But then how did you come up with this one gene idea that you mentioned, and how did that transition into an ag biotech company?
I think I could tell that story fairly quickly. Basically, I was a consultant for Lilly for several years. I worked my way up to be one of the senior consultants for several years. So it was on the ag division, and they had a lot of valuable herbicides and a lot of others. So we were a group that met once a month or quite often in Indianapolis. And when we arrived, still on Western time, I was usually half asleep, but the head of the vice president at that division had you brought in every time a person who gave you a talk on marketing. Now, I knew nothing about marketing, but after a while, I began to soak in a little bit, and when he started to use some of the numbers, and I found out that Monsanto had this compound 1538-1 that was called Roundup, and it was a $1/3 billion industry just to spray the weeds in your. So I knew that, and so I knew the value if you could make that soybean safe. And we'll jump ahead to today. 200 million acres of them, of mostly that crop. 200 million acres, a lot of acres. So it turns out, but the problem so I knew that. That was one. Number two was that instance at Asilomar when the first day Eli Lilly's group funded Genentech to build a new kind of way of making human insulin to avoid any possibility of self-immunization. It happened with porcine and horse. And they had showed how they did that, and they made E. coli into a factory that cloned and sequenced, and it effluxed humulin. It's called humulin if you want to buy it. If you have type 1 diabetes, you probably take humulin today. So they're the biggest. Eli Lilly owned the insulin industry. But they told us this simple story, and I just fell over. That's number 2.
Yeah. So they took a human gene, they put it into E. coli,
They
and
took the
they made a
human
factory.
gene, made it in an E. coli. So this gets me back to, you can see where all this is headed. So I began to immediately, from that point on, I say, this has got to be powerful somehow for agriculture. So I wasn't totally naive. As I was told at one time, and Bill will have to later on correct me on this, that there were over 100 plant physiologists here at Davis, and I think about 90 or more plant breeders, and etc. I mean, huge number of people, experts in this field. So I was here, and I was asked by the dean finally to do my teaching duties through a standard department, which was agronomy marine science. So I was expected to go to hear these meetings, go to all the, and so I didn't learn a lot about the the husbandry side of plants. I was quite interested in the nitrogen fixation part, but I realized that pest control and herbicide control was a very valuable situation. So, but I didn't know that almost all the great discoveries in plant breeding were of a quantitative nature, not a qualitative nature. And so when I proposed later to the university, I went from the meeting, stewed a while, a year or two, wrote a white paper to the University of California Dean's Office, which went to the committees of agriculture direction for the state, that said, you know, you're using a single gene approach. And the plant breeders heard about this, or whoever, said, never been done in history. But then Cal Qualls came in one day and dropped a book on my office, and it told about Burton's famous experiment in creating frost-tolerant, what is this grass that gets in your lawn and you have to cut out? Frost-tolerant, and he used the one gene approach. That was the only one in history. So, well, it's a start.
Had you identified the gene at this point that you wanted to target?
In a way, yes. It was. It took about 10 years to figure that out, and you know, it was nitrification genes, symbiotic genes for symbiotic nitrification. You'd have to have a Manhattan Project, 100 billion maybe, to do that.
Yeah, it's a big
So
pathway.
as you go from 1 to 2 to 3, we didn't know it, but it's impossible. So I'd already figured that out, had the data to show that you can't do that.
Yeah.
So you got to pick something else other than nitrogen. So we sold the idea at first on the nitrogen fixation idea, but that didn't last very long. We already knew at Asilomar that was impossible to do.
Yeah, and you had a front paper in Science where you showed the energetics and how that was not so feasible.
Well, actually, it was no, it wasn't in Science. That was on osmoregulation. It was about, this was a paper in a well-known micro journal that showed we developed a model system to show that if you want this bacterium, this Klebsiella, which we got the genes out of, to fix nitrogen, we had to make mutants that all of the energy going in as sugar came out as ammonia milk, as ammonia.
Okay, so let's going back to the gene that you decided to target. So you knew about this, about Roundup, and you chose the gene then over this 10-year process. Tell me about it.
Well, it was really based on the fact that you could, you know, get this gene, probably find it in bacteria. And I didn't know this until we started really getting down to the details, when it turns out about the time we were thinking about forming Calgene, after the university decided that this was not a priority of, it was not a sound strategy for developing plant breeding. It would not be useful. And everybody agreed to that. And I do today realize that that's why you only have about 2 or 3 of these billion-dollar genes. But it turns out one of them isn't Roundup Ready. But it almost didn't happen because it turns out it's 2 genes, one in the roots and one in the shoots. And so that cost 10 years of research. But we didn't know, we didn't bother with details like that. So we started on the basis of the simplicity of this system. And when we finally sat down on the blackboard to write the final strategies, I was responsible for the marketing, for the biochemistry, and my associate he was a good financial guy. He had all the financial.
This is Norm Goldfarb.
Norm Goldfarb. My first one was $1 billion product per year. Second, green, and you follow hybrid vigor, all 10, and we followed that, and E. coli became the system, and it turns out that Roundup is deadly for E. coli's growth. You put it pure material in a petri dish, it kills billions of them, but a few will become resistant. And in the first experiments, took about a month after we formed Calgene, we had 103 of them, and some of them are tiny, some are pinpoint, but some of them are big and healthy. So in there somewhere, and 3 of those were in the target region for Roundup.
And so these are genes that break down glyphosate, which is the active ingredient.
Not breakdown. The target, the glyphosate binding target in the aromatic pathways of amino acids is not existent anymore. You don't have it because you need those aromatics. So that was the green part. You could eat this stuff. Cows come. Well, I wouldn't try it, but you could not be.
There's still a big debate today over
it. I know, but there's no debate. I mean, but they have gotten resistance problems after 25 years now. It's cost Monsanto finally. They had to be. They sold themselves, poor guys, for $62 billion to Bayer.
So so you developed this and you spun out Calgene, as you said, which began to develop this, and then they sold it to Monsanto. Is that right? And it became Monsanto's
Well, we
primary.
sold ourselves to Monsanto. We had no choice because they made a huge mistake and switched all that money. Roger
Salquist,
the new CEO, he claims, and that's in Google. If you look at Roger in Google, he raised about $200 million for this Roundup project. He took it all and poured it into another project.
Well, so, I've read the headlines, the tomato that ate Calgene.
The tomatoes that ate Roger, ate Calgene.
Yeah, and that was a follow-on project to try and develop tomatoes that had better ripening properties.
But one thing, back to something, we went over this too quickly. We need to redo this thing altogether. What happened? I was here, and Hybrid Vigor was operating the whole time. I didn't know anything about agriculture. And certainly Dave was dead, right? I mean, that they knew about every aspect of it, right? And so it just osmosis in. So I knew that the value of this product for a lot of reasons. And there were a lot of hurdles, but many of those hurdles would not have been solved if it hadn't been happening at UC Davis. And I don't think this is well known at all to the public. And I don't think Monsanto himself wants this to know this. They completely blanked it out, and 40 other people, because we operated in a way that the original idea was developed in the plant growth lab, plain and simple, but we didn't do the demonstration. But still, that early history was right out of the school's knowledge base. And that is not known to this day. And that's probably one of the biggest messages I got to tell you here. That think about it a while. Would this have happened here? It would not have, because Monsanto, we had to go out there and give a seminar and tell them what we were doing. They had 30 people the next day thinking about this. They're not an innovative, they're innovative in chemistry. They were not innovative at all, but they had a critical tool. They had the best laboratory doing regeneration, studying totipotency in plants. They had that going. They did not have any of this other knowledge, none of this philosophy of Delbruck and blah, 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 at all, zero.
Yeah. And this was the early 1980s, and so this was a very different time in the relationship between academia and industry.
Oh, man.
And I guess that some of this, this was written up in New York Times and Nature as this all came to be, and I guess a lot of the rules and regulations that exist for the University of California today were solidified during this time. And can you talk about your transition then from developing this idea here at UC Davis with hybrid vigor and with the synergy with agriculture to the point of spinning out a functioning company, and then where did that take you?
Well, it was very interesting at that point. It was clear that there was value, a big value, there's a big market for this material. So it came to pass. And it paid, Monsanto turned out to be, their stock shot up to 135 at one point. They paid paychecks for 32,500 people worldwide, and they paid them for 20, 25 years. But there was a 10-year hiatus when it made them no money because they didn't know that the gene and the roots and shoots didn't necessarily have to be the same. That cost them a lot. But so Monsanto deserves a lot of credit in persistence and deep pockets. We could never have done that in university, I don't think. Yeah. But at the same time, I wonder if we could have done, if we would have been able to do the proof of concept, then it could have been a patent of great value for the UC. So that's the only thing that, in those days, remember the patents for DNA, that was $1 billion, Stanford alone, they have you can do the arithmetic on this one. When they DNA patent, they patented the genetic engineering general system, and that was multi-billion-dollar value, went into their programs and paid students and fellowships and everything else.
Okay, so we just have a few minutes left, so I want to talk about what you've been doing since your retirement. So you retired in an early retirement initiative program, and split your time between salmon fishing and working at Calgene.
Well, this has turned out to be an amazing period because in '92 I retired. And I wanted to go fishing for the rest of my life in Alaska, and I had the equipment to do it. And I could, if I didn't buy too big of a boat, I could probably keep it going for 30 or so. But after a few years, I got a call from, of all places, from then Calgene was basically entirely Monsanto, but still called Calgene, from the vice president there, who asked me to come by and talked to them about a project they had in mind. So in 1985, the head of Roger Salquist had fired me on the spot for insinuating that he was screwing up. He had more shares than I did. So it turns out when you
came out against the tomato. So
I come out. I didn't come
you.
out against the tomato. I said I was for the Roundup, and that you can't just pick another plum like that off. Yeah. And he thought it was going to be easy with that much money. And one event, he paid $121 million on the tomato project to one group. He bought a tomato company, all the trucks, their yellow trucks go up and down Highway 5. I still see them today. He paid it, way overpaid. And he had to sell them back for 1/3. That tells you you can't do that, all because he didn't listen to scientists. So I have these experiences, and you learn from them. You hope. So basically, what happened is this: that I got bored with. I only fished 3 months a year, And it gets cold after that. So I did that for 30 years, and I sort of got that done. But 9 months a year is open. So I decided to pick a project, and the opportunity came actually back at Calgene. From that 85 report, I said agriculture in the 21st century needs to close in between standard agriculture and preventative medicine for people and animals. Wrote the report, picked the target, and it was omega-3 genes. And so that is a very complicated process. All major industries DuPont, spent hundreds of billions of dollars trying to do this. And again, for two reasons, it has not been really successful. One, it's a lot of genes. Two, it's an admixture of genes that you do. So the chemist can't make the molecule. The people who purify fish oil, cheap fish oil, can't get it cheap enough to make the pure product, which you need for baby food. But algae have done it. And one of the ways they do it is they invented, and actually bacteria out there first, invented a new way to make the molecule, to biosynthesize it. And it's called the polyketide method, and that of omega-3 production. And it's beautiful because the bug purifies it entirely to one molecule. You're not left with an admixture. And that bug, I knew a lot about that bug. I worked on it for many years. And what happened was one experiment started this whole deal. E. coli again. E. coli does not have, it hates unsaturated fatty acids beyond one double bond per 18 per tail. And however, it will accept, and we knew this, it will accept fish oil if you treat it right. There's benefits to it. So you can remove all of the unsaturations from the strain. It's dead because it can only make saturated. You can't run a system on that. But if you give it the choice of 1, 2, 3, 4, or 5 individually, it can grow. But 6 it does not like. It kills it.
So 6 would be DHA,
DHA
docosahexaenoic
is the
acid.
king, and it kills it. And it kills plants too.
Yeah, that's 6 degrees of unsaturation, just to be clear.
6 kills plants, biggest herbicide you ever put in. So all these people, all these years, by 95, I knew that that's what industry was pouring money into. That's what they still face today. It's a super herbicide in the machine that makes it. So currently, the market is heading to $1 billion for pure DHA for baby food and all the other related
supplements and everything else.
And everything else. So it's story all over again. So make a long story short, the Japanese went with the Keiko and other machines below 8,000 meters in the ocean.
That's a submarine.
No, 8,000 feet.
No, meters. You're right.
What? No, not. That's wrong. Not. It's in feet that it starts.
Okay.
You start finding these bugs at 8,000 feet down, and then as you go down to Challenger Trench at 30,500 feet, you get a lot of them, and they have the same composition of oil as your brain. Which without it, you have no computer, you have no brain, you have no eyes, you have no sperm. You're in trouble. So that was my question, that they let me, for consulting on commercial production using this new procedure. We had E. coli strains that made a little bit we bought strains from Sagami Chemical Company that made a little bit of DHA by this new method, but not enough to tip any balance of anything. So we finally made a construct, and all the books the 3 books we've written on this, David and I have collaborated on 3 books. One experiment drives the whole system, and that was E. coli without any monounsaturation. There's a little blip as you get right down there. I wouldn't say any, but very little. It needs 50% monounsaturation under normal growth usage, 50% of total membrane. So you can replace that 50% with omega-3s, up to 5. And so the queen is 25 eicosanoic acid. And it's a structural, and it simply gives them more energy. It's an energy. It's speed. You have speed as what you need, just more speed. And you pay for speed. Speed always kills. So that's basically the story.
Yeah, they're paying for faster metabolism and more energy production.
So what this all turned out to be is this agriculture story now has morphed into the weirdest story, and I've already sort of communicated to 2 people in this room about what that story is. It's getting better every day. So what I told them a week ago or 2 weeks ago is holding up, that, you know, life in general, all life forms in general, have to have, you cannot do without energy for any process. Any process. If you want to keep a mitochondria going for 5,057 years, or a chloroplast for 5,057 years, you need...
Yeah. And that would be the oldest living organisms on the planet.
Pine, chloroplast is 5,000, but they found now one over in Sweden, a pine.
Yeah.
His chloroplast is clearly 8,000, over 8,000 years old. And Ming, in the animal world, the record now is Quahog at 511. His mitochondria, or whatever you want to say, they're viable, whatever that means. After 511 years, they didn't wear out. So what is the secret to that? And has that got anything to do with Alzheimer's or 250 other diseases that we face, including aging itself? Now, you wouldn't want to live like the bristlecone pine has to live. It is on the fringe of life, and if you take it out of that fringe, it'll die. You try to grow it in your garden here in Davis, you kill it immediately.
Well, this has been really exciting for me to get to sit down and walk through all of these stories with you, and there's so much more that I think we could still touch on. But I just want to thank you for taking the time to sit down today and doing this, and look forward to what you discover next week.